The overall goal of this procedure is to adapt and maintain human-induced pluripotent stem cells to feeder-free culture using complete KnockOut Serum Replacement Feeder-Free medium. This is accomplished by first preparing Geltrex-coated or CellStart-coated plates. The second step of the procedure is to passage your IPS from MEFs to feeder-free medium using Dispace. The final step of the procedure is to continually maintain your IPS cells feeder-free. Ultimately, results can be obtained that show similar cell growth and stemness found with MEF cultures through immunostaining with pluripotent markers.
Hi, I'm Kate Wagner at the GIBCO site at Life Technologies. The main advantage of this technique over existing techniques is that you can save time and increase consistency in your cell culture when you use KnockOut Serum Replacement throughout your entire workflow. Today, the family of KnockOut products includes a full list of media and reagents for feeder-based, feeder-free, and xeno-free conditions for human embryonic stem cells, mouse embryonic stem cells, and induced pluripotent stem cells. KSR can be used for derivation, growth, expansion, cryopreservation, and the initial steps of differentiation. This following video will show you adapting and passaging your cells using KSR in feeder-free conditions.
Geltrex-coated culture dishes are required for adapting and passaging human-induced pluripotent stem cells in feeder-free conditions. 1 day prior to the preparation of these coated dishes, thaw 1 tube of Geltrex slowly by leaving it in the refrigerator overnight. On the following day, dilute the tube of thawed Geltrex 1:100 in basal medium by transferring 1 milliliter of Geltrex to a bottle containing 99 milliliters of basal.